Everybody, can I ask you to take your seats, please, so that we can start on time? Thank you. So the first speaker this morning is Angela Hodges from King's College in London, and her title is Exploring Alzheimer's Disease Risk Variants Linked to Alzheimer's Disease. Thank you, Angela.
Thank you. Good morning, everyone. So, as Angela Hodges from King's College London. I'd like to thank the Biochemical Society for this opportunity and the organisers for allowing me to present our work today. So as you know, Alzheimer's disease is a complex disease. It's many risk factors, both genetic and non-genetic. I've tried to line all the non-genetic ones here up in terms of their effect size. So some have a big impact on protection, some have a big impact on risk. But it's complex. And in fact, probably some of the genetic factors will play into some of these environmental risk factors as well. On the genetic side, which I realise now is squeezed right at the bottom of this slide, we know that it's quite a big impact on disease overall. Of course, there are some well-established, high-penetrant mutations and genes related to amyloid processing and production. But there's a whole lot of other ones, and of course, I'm sure you're familiar with ApoE, which is one of the big effect size genes with common variants. But it's polygenic, so a whole number of these. And it depends how you do your list, but there's probably as many as 83 solid genes now from all the genetic studies over the years. But we're really interested in using particularly the genetic knowledge here to understand the functional pathways that these genes are involved in. Whilst there might be some benefit in using these genetic information to enrich populations that have a higher risk, they'll never really probably be used at the individual level unless there are these mutations in these genes for diagnostic purposes. But they can tell us an awful lot about genes and pathways and functions that put us at risk or protection. So, as I already suggested, it informs pathophysiological processes. And for quite a long time now, back in 2010, one of my former colleagues, the late Leslie Jones at Cardiff University, was probably the first to recognise that a lot of these risk genes were falling into some kind of solid areas of biology. So lipid biology, endocytosis and immunity, and of course we already knew quite a lot about amyloid and tau. And this is really underlined in this recent Nature Genetics paper here by Linguas et al. Huge effort, really large GWAS study where they've added further genes to the list, but these fundamental processes seem to be overrepresented in the genes associated with the disease. But many of these processes, of course, particularly immunity, but also lipids and endocytosis, can be linked to microglia, which is an area, obviously, we're all really interested in. And many of the Alzheimer's disease risk genes, as you've already heard a lot about some of these genes already, are highly or even only exclusively expressed by microglia in the brain, so they're not really in any other cell type, at least in the brain. So here's an example here of ABI3, rare risk variants in ABI3, and you can see some staining here in human brain tissue, and in collaboration with Jenny Pocock here, we've stained also in IPS microglia. So this is just some data taken from some public data, single-cell RNA set data. We heard already about this data, the Seattle Alzheimer's Disease Brain Cell, CAD data, lining up some of these genes in terms of their enrichment in microglia. And you can see this gene at the far end here. Pretty much all of 40,000 cells, microglia cells in this data set express SOR1, so it's highly expressed and it's pretty much in every one of those 40,000 assigned microglia cells. At the other end of the spectrum, you can barely see it here, but this GPR141 risk gene is also expressed in microglia, but at least in that data set and at the level of RNA, it's only in about 2% of the cells. So the reason why I point this out is because if you're wanting to model these risk genes and understand their function, then it will be really important that you are establishing that they even express the gene and the pathways. So that's why I sort of point that out. And then you've got everything in between. And of course, we've already heard about TREM2 and CD33 here, but nowhere near all of those 40,000 genes, at least in that data set, express it at the level of RNA. And just to highlight again for ApoE, you probably already are quite aware that it was always recognised as an astrocytic expressed protein, but you can see that as people have become aware in recent times, it's actually also really highly expressed in microglia. And then for comparison, a couple of established markers here, CD45, that's the name of the gene there, PTPRC, and IBA1, which we've seen a lot of staining for IBA1, of course, at the level of protein. This is RNA data, but just to point out, at least in this data set, that only 22% of the cells actually express this gene at the level of RNA. And then these are the ones that are more highly enriched in microglia, but then many other of the AD risk and protective genes are also expressed in more than 70% of microglia in this data set, but also expressed in other brain cell types as well. So what we really want to do, though, is try and understand the genetic mechanisms of these AD risk and protective variants. And we heard already this week, Jennifer Pocock mentioned on Monday, the TREM2 receptor. It's very high risk, high effect size gene, but it's quite rare, the mutations, and they cluster in this extracellular domain here, and as Jenny explained, it impacts their missense variants, so they change amino acids, and they impact ligand binding and cycling of the receptor up to the cell membrane. We had a great account of CD33 yesterday from Peter St. George Hislop, so I don't need to talk about that, he's a real expert, but we heard about big M and little m isoforms, protein isoforms, where the variant impacts splicing of exon 2, that's the functional SNP, and its interaction with its binding partners. We also looked some time ago at MS4A6A and established that the risk variant impacts allelic expression from the gene. So in many ways, the genetics is actually really challenging, because when you read a GWAS paper, you assume because the nearest gene to the variant is going to be the functional impact, it's going to functionally impact that gene. That isn't necessarily the case. And a lot of the variants, whilst some of them are easy because they're in the coding sequence and you can make good hypotheses and test their impact, a lot of the variants actually sit in non-coding regions and intergenic regions and introns, or even some distance away from the genes. And so you'll notice over reading the GWAS studies that some of the variants have been assigned to different genes over the years, even if it's the same variant. So investigating the genetic mechanisms can be quite challenging, but I'm going to present some recent data where we have been looking at SORL1. And just to remind you that SORL1 is really highly expressed in that CAD public data. In microglia, there is some expression in neurons, but nowhere near to the same extent that it is in microglia. But interestingly, much of the work on SORL1 has been done either in non-brain cells like HEC cells, or it's been done in neuronal cells, but I don't think there's been a lot of work in microglia so far. So SORL1 is quite complex in terms of its genetics. It is both associated with sporadic late onset AD, and in fact the minor allele is, the rare allele is, more rare allele is protective, and it was first published, I believe, from Peter St. George Hislop's lab some time ago, where they identified that there was probably two genetic features, one at the 5' end of the gene and the N-terminal end of the protein, and one in this 3' haplotype, and they did a great functional analysis of this genetically. But as I mentioned, most data since then has pretty much been generated on non-brain and non-neuronal cell models. It's a really, really large protein. It's probably one of the largest in the human genome, so it's obviously quite challenging to work with. But we know quite a lot about its different binding motifs, and it's part of the LDL receptor family. So in fact, it will be suggested that it binds apolipoproteins. I don't actually know whether there's data whether it binds ApoE, but it certainly has those features. This region, this LDR receptor class A repeats, also has been shown to bind APP, and you've got this binding part of the protein, and then you've got this class B repeats here, which releases the apolipoproteins. It's considered to be an endocytic protein, so it probably is sitting there receiving the apolipoproteins as they come into the cell. But not only is it associated with sporadic disease, but it's also been described more recently, well, not so recent, 2012, that there are rare familial missense changes that either change amino acids or they lead to truncations, and these are distributed throughout the SOR1 protein, so no particular bias towards any particular motif in there. And the binding of APP is believed to slow APP release from the Golgi, which reduces APP processing and the production of amyloidogenic A-beta. At the other end, you can see that there are these VSP-10P domains, BNR repeats, that directs A-beta to lysosomes for degradation. So the settled, if you like, SNP that's been come out of the GWAS studies is this SNP here. You can see its RS number there, and it's located in an intron pretty much in the middle of SOR1 gene, so it's intergenic. So there's no clear path then into what is the genetic mechanism that is contributed by this variant. So we set out to explore this further to try and understand why this variant might be leading to disease. And just to remind people who aren't so familiar with genetic studies, it isn't necessarily the case that the SNP that's been reported in the GWAS studies is actually the functional SNP. It will be tagging an area, but it's not necessarily the actual functional SNP. Sometimes it is and sometimes it isn't. Anyway, we did RT-PCR across various parts of the gene. So at this end, which picks up two of the main brain isoforms, which we knew about from our own brain RNA-seq data from some time ago. And we also mapped various analyses across this intronic region where the SNP resides as well. So you can see the close-up there. So we have access to post-mortem donor brain samples from Alzheimer's disease controls. We also have our TREM2 risk carrier cases in that set as well. And we compared expression levels, and there was no difference in any of most of the SOR1 products were no different between AD and control. There was no TREM2 related effect. There was some variation in primers that sat across the intronic boundary. So you can see here that there were quite variable levels across control and AD brain, but it wasn't enriched in AD or control. We also, so we, in the course of genotyping our brain donors for the SOR1 AD risk variant, it's actually a protective variant, we made a product from both RNA and DNA, and we sequenced it to establish the genotypes of the people. And to our surprise in the RNA samples, what we found is that all of the samples had a really, really high abundant transcript in this so-called intronic sequence. And you can see it here across all of the samples. It didn't really vary between control and AD samples at all, or in respect to having a TREM2 variant, or with respect to having the rarer protective allele. And it's novel. It's not described in any of the ensemble expression database that you can see here in green. Of course, most genes are really complex. They produce a whole range of different transcript isoforms, as we've heard this week already from the long sequencing, but also from RNA sequencing data. And when I looked at some recent data generated by John Mills's group on cortex, brain cortex, this wasn't included in any of the isoforms that they described, so it appears to be novel. So we set about extending our knowledge of this area, and we designed many, many different RT-PCR primer sets, and we could see expression pretty much with all of those in our brain samples, as you can see here. And it's a contiguous transcript. You can see this one here is about 1,300 base pairs with this primer and this reverse primer, and extends into this exon here, which is actually a 5' untranslated region of the 204 more minor isoform in brain samples. So it's probably longer than a typical exon. Of course, at this stage, we didn't know if it was mRNA, a different isoform of mRNA, or whether it was some kind of regulatory RNA. But we also, with the collaboration with Jenny Pocock, we established that this novel SOR1 transcript is also expressed in iPS microglia. You can see it's actually also in the earlier iPS as well, and compared it to SOR1 mRNA expression here. Sorry, it's a bit wavy with the beta-actin in these particular samples. So we set about doing 5' and 3' race, and so far we've at least attempted 3' race, but I don't know how many of you have had experience of this method, but it's actually quite difficult to get it to work in a specific kind of way. Of course, the other thing is that we are expecting our transcript to potentially be really large. I mentioned that this is a really big protein, SOR1, and if it was an mRNA isoform, the transcript could be anywhere up to 15,000 base pairs. So we did a an oligo DT priming from what we hope is a poly-A tail through to our transcript, but we only ended up with non-specific products. We're expecting something above this line because from our previous work we knew that this was at least 3,000 base pairs. However, we can detect using the more focused and smaller product primers, we can detect it across our samples. So the other thing is that we're using post-mortem brain tissue, and it is quite challenging because it has a variable RNA quality, although I was using very high-quality RNA samples from our set. We wanted to establish what strand it's expressed from, so whether it's expressed from the top DNA strand or the bottom DNA strand, the plus or the minus strand. SOR1 is expressed as the same as the top strand, and so we designed primers with linkers for making the cDNA, and then did some nested PCR. And you can see the results here for SOR1 mRNA, which we know, as I said, is the same as the top strand, that you can see that you only get a product when you have primed the cDNA from the correct strand. When we compared it to our novel transcript, you can see it also is expressed from the top strand. So this tells us that it could well be that the novel transcript that we're detecting may be an mRNA SOR1 isoform, rather than a non-coding regulatory RNA, which it could have been. I mentioned that we genotyped and sequenced the product across this novel transcript, and it doesn't appear to be that there's any preference for one allele over another, so it doesn't seem to be that the variant is an eQTL. But what we did find in the course of doing this is that not very far upstream of our Alzheimer's disease, well, it's protective, I call it risk variant, but actually this is the protective allele here, is another SNP that's been characterised and in fact has been associated with longevity in centenarians. Sorry, I can't say that word. But what we found is just a little bit upstream of that actually is in perfect LD with our heterozygotes here, is that there was this novel one base pair insertion which would potentially, if this was made into an mRNA isoform, be pushing this out of frame if it was a coding region. So we look to see whether this region of the intron has any homology with anything else, now that we knew that it was being expressed from the top strand, and it doesn't have any homology to any other sequence at all. It doesn't have any homology to the SOR1 mRNA, which might indicate that it's a regulatory RNA. we found that it has similarity with other endocytic. There's a region in the sequence that has similarity with other endocytic proteins, but are not really known, well characterised. So if this novel transcript does turn out to be coding, we've got obviously further work to do on that. Then this one base pair insertion, which is in perfect LD with the AD associated variant, would change the reading frame of the protein and therefore could be a genetic explanation for the mechanism linking it to disease ultimately. So in summary then, we've identified a novel AD risk variant spanning transcript. It isn't in the databases, it isn't in sequenced brain samples. We have our own RNA sequencing data from brain, which we haven't yet, but we will be going back to interrogate to see if in fact it's there when we look for it. Because what most people do is they map their RNA sequencing data onto the known transcriptome, and so the other data may have been discarded. It seems to be expressed from a copy of the top strand, which is the same as SOR1 mRNA. It's too big to be a microRNA. It's not obvious that it's an antisense RNA, although it may be. We've got more work to do to see if it in fact has some influence over the production of this 204 isoform. And we've identified this one base pair insertion, which seems to be in perfect LD with the AD risk variant. That other variant that I mentioned isn't in perfect LD, it's in high LD, but not perfect LD. And so we hypothesize and have further work to do now to see if this could be the functional variant underlying association with Alzheimer's disease, which then allows us to better now establish the models and the overexpression and knockdown that we might want to do. We are also have designed RNA scope probes to this novel transcript, and will be able to confirm that it's expressed and where it's expressed in the brain and also in iPS cells. But it does seem to be highly and widely expressed by microglia, but we'll be using it obviously to follow up for functional work. So just lastly then, very short list. A very talented student has been working with me on this project, and I have a really great collaboration with Jennifer Pocock at UCL. And of course, we always thank the brain donors for their families for the contribution of their samples for this work. And some of this work has been funded by an AR UK grant that Jennifer Pocock and I have. Thank you.
Thank you very much, Angela. Any questions for Angela, please? No. So have you any idea what the phenotype is in a mouse model, for example, where you have overexpression or downregulation of sort? There's such a thing?
I don't know. I'm not familiar with the mouse work. Whether there's a model, I'm not entirely sure. Perhaps somebody else might know about that.
Anybody have any experience of that? No? No, okay. Any other questions? Thank you. There's one over here.
So have you looked at the mutations, missense mutations that appear to be in SOL1 and see whether they actually affect this gene as well?
No, this is quite recent work, so that would be obviously a really great thing to follow up with, but we haven't done that yet. So yeah, no, that would be definitely on our list to do. Thank you.
I think we're all a bit stumped by the beauty of it, and the novelty of it. Thank you very much, Angela. That was a lovely talk.